Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Heiko Runz, who is a leader at the cutting edge of genetics, neuroscience, and AI-driven drug discovery. Today, he's the VP of Neuroscience at insitro, which, if you're a longtime listener to the podcast, you will have heard me speak to the founder and CEO, Daphne Koller. Prior to this, Heiko spent some time at some amazing institutions, including Merck, Biogen, and I think you started at Heidelberg, right? Your background weaves together medical genetics, drug discovery, therapeutic development, and we're going to cover, I think, a lot of ground today because I've known you for a long time and have read and respected a lot of your work. So I'm really glad to have the discussion. Thanks for joining us.
Thanks very much, Patrick. Really nice introduction, and it's a huge pleasure for me to be here.
I would love to just hear about what got you interested in medical genetics in the first place, and also maybe the first step into applying that into therapeutics development, where you spent most of the last few decades.
Very happy. I can take a couple of steps back. It's been quite a bit of a journey because I've been a bit all over the place. So I specialized, in fact, in medical genetics in Germany, which involved really several years of clinical genetic counseling, diagnostics for molecular genetics, cytogenetics, really then also 2 years in a clinical setting, pediatric setting, where I was mostly dealing with new treatments for rare diseases primarily. And I think this really gave me this strong feeling about, well, how exciting clinical genetics is, what is the breadth of genetics? Yeah, we had so many different diseases from rare to common that all had various degrees of genetic components. And at that time, we became much, much better with diagnosing those diseases. Yeah, different molecular genetic tests came up. At some point, exome sequencing started to become more and more routine, and we were able to increase the diagnostic rate substantially compared to what was in place when I very initially got in touch with genetics during my medical studies. But also, you felt that in a way we were still keeping it at the level of diagnosis primarily. Right. And since there were only very few exomes generated at the time, we just had to learn how many variants you identify out of a sudden that you cannot make really entirely clinical sense of. Yeah. And I think this was one motivator that brought me, on the one hand, really getting excited about testing such variants and gene function in cells and defining phenotypes that would be closely monitoring the diseases that we were dealing with. And I set up an experimental lab at EMBL Heidelberg and the Heidelberg Medical Clinics to test at a higher throughput and on microscopy-based platforms how as RNA knockdown of such genes would impact disease-relevant phenotypes in cells. Now, that still was not entirely convincing and sufficiently close to the patient. And I felt with the really start of more and more exome data becoming available, I really wanted to learn how to make the most sense of those data. And I had the opportunity to spend some time at the Broad Institute in Boston, where I really then learned about the potential and pitfalls of those large data sets. And really, well, as life goes, we as a family got stuck in Boston a bit and excited about this.
Not a bad place to get stuck.
Environment here, definitely not. Yeah, we had huge opportunities, and it's a very exciting and stimulating environment, and still is. And my wife got a very good role. She's a scientist as well. And then I took the opportunity to go to Merck, where Robert Plenge at the time had started a new department, and that was entirely based on this hypothesis or observation that genes that have that we're pursuing as drug targets that have a solid genetic hypothesis, has a solid genetic basis, have a higher probability of success. And this is in fact really where we have been making, I would say, the first inroads across a broad range of therapeutic areas, testing how well this would work and how we can make these hypotheses translate into drug discovery programs. And in a way, this was then continued when I moved to Biogen, very much with a focus on a range of neurological diseases, primarily neurodegenerative diseases, from rare diseases such as ALS to more common diseases such as Parkinson's or Alzheimer's disease. Yeah, and all of this together really brought me into this opportunity to bring in genetics really across all aspects of the drug discovery process, from the earliest activities to nominate new drug targets to really clinical use of genetic data to help improve the success rates of clinical trials and accelerate also development timelines.
So most people listening to this podcast are familiar with the statistics you raised about genetically supported targets being much more likely to succeed. And I know that in all of your roles, you've leaned pretty heavily on population genomics, biobanks to make some of these discoveries initially. But I get the sense that your work has gone a lot deeper beyond just, hey, this is a loss of function that's interesting, but how do we connect it to biology? How do we use these biobanks for much more than just making that initial discovery? I'd love if you could talk a little bit about some of the things beyond people's first impressions of how these biobanks are useful in therapeutics development.
Yeah, thanks very much. Fully agree. This is one of the most exciting aspects of my own work, and what I feel has really now become possible over the last recent years with the substantial growth of those biobanks. Yeah, we had the opportunity to participate in the exome sequencing of a UK biobank, and with this really generated now exome data at such a scale on individuals that had been profiled with this amazing UK biobank effort so deeply across a very broad range of phenotypes. In the meantime, I was also in a position to help lift off the FinGen consortium, where we were really bringing together multiple different pharma partners and a set of biobanks in Finland to help generate genetic data and link this into this amazing Nordic health records that very deeply characterize patient and population trajectories over long periods of time. And I think one of the key ideas was very early on, how can we really make the most of those data sets by not looking just on one of those biobanks individually, but really trying to increase the power by making merges between those. And UK Biobank and FinGen, we had the opportunity relatively soon to really make matches between phenotypes that have been captured and have been, aligned relatively well between those two biobanks, and then use those phenotypes for improved association testing. And together with a very detailed genotype data in FinGen and exome data in UK Biobank, we were able really to bring this at the level of individual coding variants or conduct gene burden tests that then really led us to individual genes that were strongly associated with phenotypes, broadly of relevant to health, but also phenotypes that we were concretely aware are reflective of diseases in need for new treatments.
There's maybe an interest. We've probably got a couple of interesting stories. You can pick the best one, but one that springs to mind for me is the relatively recent publication, I think in 2024, of CFHR5 loss of function that looks to be protective for age-related macular degeneration. These loss of function variants are wonderful when you discover them, right? Because they become very natural drug targets. Can we safely inhibit this gene in everyone or knock it down in people who don't have the naturally occurring loss of function? I'd love to hear a little bit about that story. Like, how was it first discovered? And then how did you actually chase down the biology and what was going on underneath that?
Yeah, this was actually a really interesting discovery, and the path to where we are now has been taking several years. And I think I personally took so much learning just from this particular case. It came up exactly in this type of study that I described earlier. The merger of UK Biobank and FinGen, a single coding variant information, where we found that a frameshift variant that was substantially enriched in FINS was in the CFHR5 gene was protective for age-related macular degeneration. So age-related macular degeneration is a very common form of blindness that affects about 1 in 5 over 90-year-olds. So is a fairly common disease with a fairly high genetic component. And we have, as geneticists, a long-term, familiarity with this disease, just because the very first disease-relevant GWAS locus was in fact identified for macular degeneration. This was in 2005 by
I didn't know that.
Al. And at that time, they really did a GWAS that was based on just 96 cases and 50 controls. So you see it was an extremely strong association signal. That means an association signal that is long time out. And the CFHR5 gene now directly falls or overlaps with the locus for the CFH signal. The complement factor H gene is a gene that encodes an essential component of the complement pathway, so a pathway that has key roles in immune response and host defense. And we know also really very well the biology about the relevance of this complement pathway. We have now, with the massively increased GWASs that have been conducted since this very first one, discovered really several other components of this complement pathway that also provide additional genetic support that this pathway is really relevant for this disease. And there's even 2 approved treatments that are modulators of components of this pathway that are now broadly available for the treatment of especially the dry form of age-related macular degeneration. So for CFHR5, we were now in this situation that we had identified a protective frameshift variant in a potentially interesting pathway and a mechanism that we would be able to downregulate with a therapeutic. But it was right next door to this CFH gene that is very well established to be relevant for this pathway, and that just might lead to a false positive signal in this neighboring gene. So what we really went into is really dissecting the details of this particular GWAS locus through multiple rounds of fine mapping and conditional analysis that in the Finnish population is relatively straightforward because you have fairly long haplotypes and you have a fairly homogeneous population. And here, through these really deep dive on the genetics that was available, we were able to nail down these association signals to essentially just 2 genes, CFH and CFHR5, while there were several other genes in the neighborhood that could also have been a culprit for association signals. And in fact, this is a region that is also particularly under evolutionary influence. It harbors multiple different large CNVs and deletions that also have been related to various sets of diseases. And we really considered those as well and could largely exclude in this case that these large deletions have a particular influence on AMD. And we really also here went into some amazing new data sets. For example, by teaming up with scientists of the 1000 Genome Project, we were able to really do base level resolution of this locus in the 1000 Genomes cohort with long read sequencing data. And this really showed us indeed the only explanations that we have for these strong signals are coming just from those 2 genes, CFH and CFHR5. The story didn't really end there, just to tell us, well, there's this GWAS locus is explained by at least these 2 genes or 2 signals. But we also wanted to really see, does CFHR5 and the genetic variants that we identified in CFHR5 have a functional impact? And here now what I find still is a key part of that new paper is that we established a process how we could do sample recall studies from this resource. As listeners may know, FinGen is a collaboration of not only pharma partners but also 9 different biobanks that are spread across Finland, and they have samples banked from really a large majority of the FinGen participants that are in principle consented for secondary research use and accessible via those biobanks. So really by putting together the deep health information that is available in FinGen, we were able to define a distinct cohort as we felt might be most reflective and informative for AMD. We were able to use the genetic data in FinGen to enrich this particular cohort for carriers of this enriched frameshift variant in CFHR5. And in principle, with a fairly long process to get access to those samples, we're able to ship those samples to a variety of labs who generated proteomic profiles because we wanted to learn if this frameshift variant indeed impacts CFHR5 or also other proteins, and to conduct functional analysis of the complement pathway. And indeed, this worked out then really beautifully. We could validate those genetic mechanisms through and demonstrating an impact on protein level, and not only CFHR5 itself, but also functionally related proteins that indicate that loss of this gene really has a broader impact on how the complement system works. And then demonstrate also that, as we would expect, a reduction of this gene, at least through this frameshift variant, is reducing complement activation, and with this, a nice template for eventually generating therapeutics that would have the same effect and ideally be beneficial for AMD.
This point about being able to recontact out of FinGen, do you run into that as an obstacle in many of the other biobanks you work with? I know the new generation of all of us and our future health are investing a lot in this, but UK Biobank, for example, is very hard, if not impossible. How important is that? Like when you think about biobanks, there's the big data sets that you and others have been helping pay for to generate these exome sequencing through these major consortiums. But then there's the on-demand recontact that can be a total linchpin in some of these kinds of discoveries.
Well, I'm still thinking here a bit as a clinician, and I'm thinking about really wealth of data that is there. And I think the field has a bit underutilized the opportunity yet that these biobanks provide beyond the broad scale association testing and just making these data sets larger and larger and larger. I fully understand that there comes quite a bit of operational complexities when you want to go after running many of those studies from these sample collections, just because these samples are being limited and fairly precious. And in the case of UK Biobank, for example, it's absolutely understandable that you would want to primarily run the same set of assays in a large number of individuals, just as we've seen and also been able to participate in the UK Biobank proteomics project, for example. The same is probably true for most other databases as well, biobanks as well. So in this case, really the opportunity to be able to recall patients for dedicated donation of samples or increasing the availability of biospecimen is something that I feel would really benefit many of those initiatives. And I think there's always then the trade-offs, about really initiating such a study with all the complexities, because of course you have now a fantastic data set where you can essentially pick together the phenotypes and the participants for such a trial and define such a trial really well based on the wealth of data that is available. And this is really then opposed to, just generate data at scale. And in this case, really, if I had now a time machine, would go back. I'm not exactly sure if I would have initiated this recall study. Overall, really getting all the permissions and making sure that all the samples could be shipped internationally exactly to those places and the data was generated. This was clearly the longest part of this paper. It took almost 2 years. And in the meantime, there was, the proteomics data becoming available in UK Biobank. FinGen itself had profiled a subset of participants with proteomics data. And in fact, we've now really put both of those in the paper. And you could see that just with scaling up data generation, you could probably get also 80% to where we got with this very dedicated profile.
Yeah, that's interesting.
Really, so you're either back and forth. And in our case, we got, of course, now functional information for assessing the complement pathway, which you would not be able to ascertain at such a scale. And I feel for these type of insights that are really more targeted, more disease specific, it is a huge asset. And I personally would be very excited if there was more biobank initiatives that would try and make this possible.
Yeah. You've worked at companies of quite different scales, and I'm really curious to get your perspective on, not everyone has the resources of Biogen or Regeneron or AstraZeneca or some of these organizations that have, a huge team of scientists that are working on some of these major data sets. And you're not at a tiny biotech today, you're at a company that's somewhere in between. And I'm really curious to to get your sense of, like, you can't do everything the smaller that you get, and you have to focus on your core strengths, and maybe you don't have the computational resources to be the first one to discover the target, but you have some unique and scalable way to do phenotyping or something like that. I'm curious to get your take on how people in different sizes of organizations can think about using genetics at scale, because not everyone can compete at the same thing, right? You have to pick your battles.
And I mean, this is true for any size company, I would say. Yeah. Also for the larger companies like Merck or Biogen, the key focus has to be on generating medicines that differentiate from what's out there in the market and help patients ultimately. So the true focus is really on the drug discovery, drug development side, the target discovery side that most of our field is really focusing in is the nice to have. Yeah, it's shifts the baseline, and we very much hope that the overall field will really follow our hypotheses and our findings and our confidence that this will make the better drugs. But it is always a bit of an uphill battle to really try and revolutionize all these set paths that we have in drug discovery and drug development, mostly with targets that we have just no clue about. We have a strong statistical association. We have confidence in the target with the sort of analysis that I just explained for CFHR5. We can become more and more confident. But that is still often not explaining a huge chunk of biology that traditional drug discovery is being comfortable needs to be comfortable with to actually start working with the drug discovery pipeline. So there's still quite a bit of a substantial gap, and I feel a lot of researchers that I mean there's a huge space, I would say, that to fill that gap with smaller labs that may not have the computational resources to contribute to major data generation, but that could really start diving into those data sets and pick their pathways or diseases and start really bringing hypotheses further away from just the statistical association to a mechanism of action and function. And this is certainly an area where biopharma is not necessarily incentivized to spend substantial time on. In our environment, it is always we need to start a drug discovery program as fast as possible, and we need to bring it into the clinics as fast as possible. And the more confident we can be with a genetic hypothesis, that's great. Maybe we can cut corners in some ways and bring it faster into preclinical activities. But mechanism of action is often not as deeply looked at as we would wish to. And in fact, this is one of the reasons also that brought me to insitro, where insitro has been really very much putting emphasis on generating such platforms that drive the best possible science, and not only on amassing a population scale data that help us to generate the best possible targets and help us with really evaluating what bets we should make ideally as a fairly small company, but then also really putting in place cell-based high throughput assays that allow us to monitor disease-relevant phenotypes in cells and evaluate multiple different possible targets against each other so that we really have means to bridge this path from statistical association to function.
Yeah, in the context of neuroscience, what is the current state of the art and the edge of the field right now in cell-based assays? Because it is a hard organ to model in cells, but there's some impressive organoids and single cell models out there that I know you work on, other people in the field do. I'd love to get your perspective on that.
Yeah, it's a highly dynamic field, I would say, where we have multiple different models now emerging that are fairly exciting. Yeah, iPSC-derived neurons and other CNS cells are getting more and more easy to be cultivated, and we can now scale them up really to conduct large-scale functional screens with CRISPR, with siRNAs at a genome scale almost. Yeah, so in principle, the technologies for these are available, and insitro has made quite a bit of mileage here to do this. There's, of course, as you say, organoids and other models that help us to get further confidence for some of those targets. Often that requires still quite a bit of additional work, and you would need to have the clearly customized usages. And that is always really, it's a very, very exciting field. And I'm really personally excited to be part of this and learn what of those new models that are becoming available are ultimately really useful for us to generate and validate drug hypotheses, and which ones might be really academically interesting and help us understand more of the CNS, but a bit out of scope for us and the drug discovery process.
You spent a lot of your career working in ALS in particular, and I would be remiss not to ask you a little bit about the tofersen story and also like where you see the field going from here. But for people who aren't aware of all the great work that you and colleagues did at Biogen and that continues today, I'd love to hear a little bit about what you learned from that, incredible success that you all had bringing first therapeutic to market in the genetic subtype SOD1, but also one of the most, I think, innovative prevention or early, I call it early intervention trials focused Atlas that's focused in SOD1 carriers that are pre-symptomatic, but were in the process of phenoconverting, and you have a good shot at maybe halting progression early. I'd love to just hear a little bit about your contributions and the story behind that.
Yeah, ALS is a fascinating disease with a massive medical need, as you know, given that it's a fairly common rare disease of the rare and neurodegenerative diseases that you would want, that are desperately in need for treatments. And at Biogen, I had the privilege to participate really in a very customized therapy development effort for a small slice of the overall ALS population, SOD1 ALS. That means individuals who are carrying pathogenic variants in the SOD1 gene, and with this showed a specific form of the disease is clinically very difficult to differentiate from the rest of ALS. So in this case, really, a genetic diagnosis is a very critical component of identifying those individuals. And my colleagues at Biogen, and together with academic collaborators and Ionis, had been making substantial mileage on generating an ASO that specifically was able to tackle SOD1, a protein, the SOD1 target in the CSF. So we could really get confident with the first clinical trials that this might be a treatment that might really be beneficial for SOD1 patients. And we were at a stage when I joined Biogen at the time where we had to design phase 2, phase 3 clinical trials and first find the mechanisms, how to identify patients with SOD1 variants, and particular SOD1 variants that would be giving us confidence these patients are progressing in a manner that reduces the variability that we would see in a clinical trial. I think one of the key contributions that genetics could do, and my team at Biogen, was to really identify patients that are in genetic variants in the SOD1 gene that were reported with a relatively fast progression of disease versus those that were going along with a more attenuated progression of disease. And this helped really, on the one hand, analyzing the data from the earliest clinical trials and help us design an outcome trial that was then powered for patients with a faster progression. Now, in the end, genetics had a relative, apart from really this trial design, genetics had a relatively small contribution because in the meantime, the biomarker field made just amazing progress, and also colleagues and others were able to identify neurofilament light as a fairly strong marker of progression of ALS. And now this marker is being fairly regularly used in identifying patients, not only where they stand along the disease, but also if they will start to develop symptoms when they are at a particular genetic risk. And as you already mentioned, there are trials ongoing that are using NFL now as a marker for ALS manifestation. And the hope is that tofersen and other treatments will be particularly efficacious if we start treating patients at their earliest stages of disease, ideally even before symptoms start manifesting. And I think this is a paradigm that we would be overall very hopeful we could expand to multiple other neurodegenerative diseases, because the general insight is the earlier we treat, the more CNS we can save, and hopefully the longer respective patients will stay healthy.
Yeah. I know you're doing work with Bristol Myers Squibb at insitro. I'd love to hear a little bit about your vision for neuroscience at insitro. What are the kinds of things that you're excited about over the next couple of years?
Yeah, so this is now a particular collaboration that addresses the whole rest of ALS, so to speak. Yeah, SOD1 ALS is a particular subform, while the common denominator for most of other forms of ALS is the mislocalization of TDP43, which is a pathogenic hallmark, you can almost say, for ALS. And together with researchers at Bristol Myers Squibb, we have been conducting screens that are assessing exactly the localization of TDP43. And we were using large-scale cell-based profiling studies to identify and narrowing down drug targets that could be beneficial to be, well, modifying cell-based phenotypes, and that have now really progressed further into drug discovery. So we are initiating the first drug discovery programs based on those findings from large-scale phenotypic analysis in cells, which is very exciting. And in a way, the hope is that we will be able to find such translatable cell-based phenotypes also for other neurodegenerative diseases. There's a number of them that in principle exist, that are in principle scalable, and that we are hoping we can run then also for a number of other diseases with similar needs, where we now can really generate a very comprehensive lists of potentially interesting targets as they're coming out from our large-scale population efforts, and that we are now in this hard situation to pick which ones do we really want to take forward into drug discovery.
I'd love to learn a little bit more about this idea of cell-based phenotypes. So and how do you actually, putting your medical geneticist hat on it, as something like this gets closer to the clinic? This is a new way for potentially patients or doctors to think about it. Maybe you could break that down. What's a cellular phenotype in a way that, might hit the medical system, and doctors and patients would start to think about this?
Yeah, I'm happy to go back, in fact, to my academic research then, where I was more an experimentalist than one who was working primarily with medical or computational information. So at this point, we were looking for phenotypes that were well reflective of disease. And one area where we have a nice bridge between cellular function, biomarkers in the blood, and clinical endpoints is lipid-related disorders and cardiovascular disease, where we have LDL particles, where we have a clear elevation of LDL that is immediately translates to elevated risk for cardiovascular disease. And we know that a reduction in the uptake of LDL particles into cells are essentially underlying this. Now you can label LDL particles with fluorophores and can conduct a very easy phenotypic assay where you measure the function of the LDL receptor by fluorescent uptake of fluorescent labeled LDL.
Right.
And now you bring this really to a scale where you measure this assay in a 384 well plate or a large scale analysis system under a high throughput microscope, and you conduct CRISPR or siRNA screens in this setting and then identify genes that are modulating this.
Right.
You do not need to stay at the gene level. You can do this also on an individual at the genome level or customized gene set level. You can even bring this down to an individual gene level where you introduce various different genetic variants, and you can really use that sort of assays to test which of those variants are the most relevant ones, which ones are considered pathogenic versus which ones are more likely to be neutral. You can use those functional assays to do combinatorial screens, for example, knocking out or down multiple genes in parallel, and with this test really additive effects. And here, I'm very excited about this as a new, not actually so new, but a tool where you can go in multiple different ways to test genetic hypotheses and, look for modifier screens, look for clarification of genetic signals that we get now really more and more routinely from the large-scale data analysis. And the same thing can actually also be done really with rare diseases. We had a similar activity where we had focus on lysosomal storage diseases, or focus essentially on one cell organelle that could be very well monitored with microscopes, and essentially looked at what genetic knockouts, knockdowns impacted lysosomal biology. And I think this is the type of assays that can really be established for a broad range of diseases, and an area that has not been explored too broadly yet, in my opinion.
Interesting. As we wrap up here, there's one more very broad topic I'm curious to get your perspective on, which is AI. It's something that insitro has been involved in before it was cool, dare I say, going back many years ago. But we've had a renaissance in the last couple of years where everyone is thinking about how do we use large language models and other kind of AI innovations in drug discovery. I'm really curious to get your perspective on where the real meat of this is. Where is the real power of AI in drug and therapeutics development, and where is maybe something that's kind of interesting, but to you is maybe a distraction from where the real exciting pieces are?
Oh, no, I must say, actually, this is a very exciting time. And for me, place to be. You have already mentioned that you had earlier conversation with Daphne Koller, founder of insitro, who already 2018 really came up with this idea. How can we help bring better drugs faster to patients, really with the help of big data and AI? And this is really a paradigm that she has been holding to over the last 7 years of the company life that has really then triggered this establishment of these amazing platforms that we can build on. And a lot of this is not just amassing the data from population resources or generating additive data in cells and through screens, but really also the best possible use of those data sets by advanced machine learning tools. And this is where I'm really privileged to work with and learn a lot from machine learning scientists in our company that help us really to refine phenotypes in both cell-based analysis as well as population resources that can serve as proxy phenotypes for the disease, and that probably we wouldn't have come up with just the classic of interpreting clinical files or measuring organelle diameters,
Yes.
as we have done in the past. And I think this is a particularly exciting time because we are really learning about the potential of those. Yeah, how much do we need to train new models based on our pre-existing knowledge? Yeah, what do we need to really go into particular phenotypes that we know are disease relevance to start training those models, or non-supervised models, maybe the even much better ways to find something that is relevant? And then how do we convince ourselves of that what these models provide is really truly relevant for the phenotypes and strong enough to build drug discovery hypotheses and programs on? And that's really a very exciting time where I feel insitro has accumulated an enormous amount of knowledge and experience, and we are hoping that we are making here the best possible bets and can come up with some very exciting new treatments in the near future.
Yeah. As we close out here, do you have any advice for people who are early in their career that are thinking about how to find their way in genomics or therapeutic development more broadly? I'd love if you could share that.
Yeah. I mean, as you've probably became clear during that podcast, I've been hopping around quite a bit, not only across continents, but also really across multiple different fields within genetics. I was very lucky and privileged to have excellent mentors that have helped me get familiar with a new field and become comfortable with a new subdomain of mine. I think really recommendation would be never lose your excitement about really learning something more. Don't ever feel too comfortable in your own field, but really search for what's missing and try to follow up on that. As genetics has become such a broad and amazing field, there's so much to discover. Stay curious and don't be afraid to take a step away from where you've started from, and be open for what you can learn, what life brings.
I think that's great advice for anyone at any point in their career. I've heard before that it's good to push yourself every 10 years or so to just try to reinvent yourself, right? Do something that pushes you out of your comfort zone. The way we are naturally, you probably won't, but you probably land somewhere in the middle between going into an entirely new field and staying comfortable. But that hopping around can just push you to learn something new and bring some of your perspectives to a new field that's a little different from where you are today.
Absolutely.
Well, thank you for spending the time with us today. I really appreciate it and look forward to speaking to you again soon.
Thanks so much. It was a great privilege.
Thanks, everyone, as always, for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.